Hi everybody, I would like to present the project Flegme for fermentation of vegetables. It is a citizen science and multi-actor project. It is founded by two French regions, managed by Vegepolis and scientifically coordinated by INRAE. So, what is fermentation of vegetables? To prepare fermented vegetables, you need crude vegetables, generally grated, salt, eventually water, time, and that's all. How does it work? Fermented vegetable manufacturing relies on a spontaneous fermentation by microbes, mainly lactic acid bacteria, naturally present on the vegetables. Bacteria eat the carbon sources of vegetables, mostly sugars, and produce lactic acid, which leads to a natural acidification of the product, and thus pathogens and most spoilage microorganisms are unable to grow. By this way, the vegetables are preserved for a long time at room temperature. Let's see the context of this project. The economical impact of vegetables is quite high in the two regions involved, Brittany and Pays de Loire. You may know that food losses from post-harvest to distribution exceeds by 20% for vegetables, and that the vegetarian and vegan market has increased by more than 20% in the previous years in France. Fermentation is an ancestral way to preserve foods. It is applicable to all food resources, from milk to meat to different types of plants. It is a sustainable process. There is no need of intricate supplies. It is available also at a domestic scale, and the product is safe without additives. Now, consumers want to renew the way of vegetable consumption, retrieve food diversity, eat lower processed foods, act for better health, and discover new taste. In this context, there are many economical and scientific challenges and also citizens' concerns. For vegetable growers, fermentation would lead new ways for production value enhancement. It could decrease food losses and also promote ugly or flow vegetables. For vegetable transformers and restaurant chefs, it is a way to create innovative products with fermented vegetable used as raw material for new recipes. It is a way also to promote the discovery of lacto-fermented products, poorly known by consumers, and to promote new flavors. For researchers, it's an opportunity to investigate the microbial diversity involved in these spontaneous fermentations. It will allow the opportunity to create a reference collection of bacteria involved in lactofermentation. We will also check domestic production safety. We will identify biotic and abiotic factors shaping microbial communities and investigate culinary uses and consumer perception. And for citizens, to get new taste, to get natural, safe, and sustainable products in their plates, and for people that do it yourself, to better understand and control the process step. The questions that Flegme Project want to answer are the following. Which bacteria species constitute the community? Is the composition of bacterial community influenced by the type of vegetable used, the addition of spices or herbs, the age of the product, the making process, for example, the percentage of added salt or the temperature of fermentation? Are spoilage or pathogenic microorganisms present? Which micronutrients of interest are present in fermented vegetables? At which concentration? Are consumers ready to accept the particular acidic taste of fermented vegetables and in which form? The main goal of this project is to provide the necessary knowledge in a well-documented form for the fermentation of vegetables, and in particular, the microbial community involved, with particular attention also to their nutritional interests, their sanitary safety, and also the creative diversification of their culinary uses. Another goal is to identify, isolate, and preserve the diversity of bacteria associated with fermented legumes, and to make it fairly accessible for the scientific community. The Flegme project began at the beginning of this year, in 2020, by the constitution of a community of people that we called fermenter citizens. That means citizens that prepare homemade fermented vegetables at home. They accept to answer to surveys on their fermentation practices and to provide some samples of homemade fermented vegetables for analysis. In lab, these samples are currently characterized for their microbial community associated and also to constitute a reference bacteria collections. Other analyses, for example, micronutrients, will also be conducted, and culinary uses and consumer perception will be investigated too. All the results data obtained will be integrated, and a good practice guideline will be also freely available at the end of the project. Of course, there will be a scientific dissemination and mediation for citizens of the results of this project. Flegme project gathers 25 stakeholders, including academics, schools, SMEs, culinary journalists, and others. The current community of fermenter citizens has more than 2,050 members. It is a multidisciplinary project, and we are sure that Flegme will favor interaction and collaboration between citizens and scientists for empowerment and appropriation of scientific approach and results by the citizens and of citizen problematics by scientists. For more details, please contact flegmecontact@inrae.fr, and thank you for your attention.